Welcome back to Chapter 15. In this second video, we'll explore how microbes actually cause disease, and this is known as pathogenesis. By the end of this section, you should be able to 1. Summarize Koch's and molecular Koch's postulates and their limitations, 2. Explain pathogenicity and virulence in terms of infectious and lethal dose, 3. Distinguish primary and opportunistic pathogens, 4. Describe the stages of pathogenesis, including exposure, adhesion, invasion, and infection. And then finally, identify examples of portals of entry and exit. For most infectious diseases, the ability to accurately identify the pathogen causing the disease is a critical step in finding the right treatment. So we're going to start with Robert Koch, one of the founders of medical microbiology, and in the late 1800s, he proposed a systematic way to link a specific microbe to a specific disease, and now we call this Koch's postulates. So the 4 postulates are that 1. The suspected pathogen has to be present in every case of disease, but absent in healthy hosts. The second postulate is that the pathogen must be isolated and grown in pure culture. Then, when a healthy subject is inoculated with this pure culture, it must develop the same disease. Finally, we have to re-isolate the same pathogen from that new host. So this framework really helped to establish what we now call germ theory and guided early microbiology. But there are some limitations. So there are some microbes, for instance, that only cause diseases in humans. For example, in HIV, we have no animal model. Also, not all pathogens can be cultured in the lab. Like viruses, you need some kind of bacterial host for some of them, or animal cell host or tissue, or even Treponema pallidum that causes syphilis. Some people also carry pathogens asymptomatically, right? They don't have any symptoms, and not all hosts are equally susceptible to disease. Our book also has a nice table covering those 4 postulates again. So Koch's postulates tell us that the pathogen has to be found in every case of the disease, but not in healthy individuals. You have to be able to grow the pathogen in pure culture, and then we're going to take a new host and infect them or inoculate them with a pure culture. So that means you're going to introduce the microbe into that healthy subject, and then they must get the same disease. And when you look at their blood or tissue sample, you have to be able to re-isolate that same pathogen. And here's a visual format of those postulates. So we have to have the suspected pathogen be found in the disease organisms, but not in the healthy individual. So you can see there is no causative agent there, whereas we have isolated it here in pure culture. Then if we take this pathogen and inoculate or inject that into a healthy host, the host must get the same disease as the original diseased organism. And then when you examine their blood or tissue, we have to be able to isolate that same pathogen. But for Koch's postulates, he makes a lot of assumptions in order for the postulates to work. And these are not always true. So his assumptions include that the pathogens are only present in individuals with the disease. And this neglects asymptomatic carriers that might be harboring that microbe but not present any symptoms or noticeable signs. Koch's postulates also assume that all healthy test subjects are equally susceptible to the disease, but that also is not true necessarily, since we vary, or hosts vary, in the strengths of their immune system. They also have different types or combinations of normal or resident microbiota that can compete and might even help you prevent infection. Another assumption is that all pathogenic microorganisms can be cultured, but that's not necessarily true either. Viral pathogens can be difficult to culture since they require specific host organisms. And the bacterium I mentioned earlier, Treponema pallidum, that causes syphilis, is very difficult. We can't really grow this in lab because Treponema doesn't make a lot of the enzymes necessary for growth, and it has to scavenge very specific host tissues and host nutrients in order to grow. Also, the last assumption is that all pathogens can infect an appropriate non-human host, but we run into issues with pathogens such as HIV. Human immunodeficiency virus only infects humans, and we cannot apply Koch's postulates by infecting healthy humans with HIV, right? So we cannot test or run Koch's postulates on these types of diseases. And since Koch's postulates does involve animal testing, I just wanted to give a really brief overview. Animal research is very strictly regulated under federal law and institutional research policies to make sure that the animals are treated humanely. So every university, for instance, that uses vertebrate animals, we have to have an institutional animal care and use committee. In the labs that I used to work in at UCLA, for instance, there were always at least 2 veterinarians on site 24/7, and we all had to complete a lot of thorough training in animal handling, ethics, and anesthesia and euthanasia procedures. Our facilities were always regularly inspected, and we always were respectful to the animals. We had to follow these principles where we replace animals with alternative methods whenever we could. We reduce the number of animals to the minimum required, and we would refine our procedures to make sure we minimize pain and distress as much as possible. So we take animal research very seriously, and we only undergo animal research when it's necessary in order to move to the next step to help humans. This checkpoint asks us to briefly summarize the limitations of Koch's postulates. So some pathogens are found in both healthy and sick individuals. For instance, Helicobacter pylori, some individuals can be asymptomatic carriers, while others will present with a disease. Another limitation is that not all pathogens can be grown in pure culture. Viruses, for instance, need some kind of host cell or tissues. Another microbe is the one that causes chlamydia. So bacteria that cause chlamydia, they can't make their own ATP. They are intracellular parasites, so you would need some kind of host. Individuals also differ in their susceptibility to disease because of variations in their immune systems or their normal microbiota. And finally, some diseases can't be replicated in animal models, and human testing is unethical. As microbiology advanced, scientists refined Koch's ideas to identify specific genes that make a microbe virulent, and this gave rise to the molecular Koch's postulates. So these postulates say that a gene or a gene product should be found only in pathogenic strains. And if we disable that gene, if we inactivate that gene, this should restore the pathogen's ability to cause disease. And a good example is E. coli 0157H7. This one acquired a Shiga toxin gene, and that turns a harmless intestinal bacterium, normal E. coli, into a dangerous one. If we knock out that gene, then the strain becomes less virulent. But if we add that gene back in, the virulence returns. And this genetic approach allowed us to pinpoint, or still allows us to pinpoint, how pathogens cause damage. So it still has limits as well, since not all microbes can be manipulated or tested in animals. So just as there are limitations to Koch's regular postulates, there are also limitations to molecular Koch's postulates. Not all pathogens can be genetically or molecularly manipulated. One example is Mycobacterium leprae that causes leprosy, can't be genetically modified because we can't grow it in artificial media. It only grows well in armadillos or mouse foot pads. Another one is Treponema pallidum that causes syphilis. We said we can't really grow it right. It's fragile. It doesn't really grow well in artificial media. Also, some diseases don't have suitable animal models, again, like HIV. HIV only infects humans, so there's no standard animal model that can reproduce the full spectrum of AIDS symptoms. So this makes it impossible to test certain virulence gene knockouts or satisfy the molecular Koch's postulate that requires disease reproduction in the host. This next checkpoint asks, explain the differences between Koch's original postulates and the molecular Koch's postulates. So in the original postulates, we're identifying a microorganism as the cause of disease, whereas in molecular Koch's postulates, we are identifying specific genes or virulence factors that make an organism pathogenic. For example, E. coli strains become disease-causing when they acquire toxin genes, like we saw in E. coli 0157H7. Next, let's define two important terms. First, we have pathogenicity, the ability of a microbe to cause disease, and virulence, which is the degree of pathogenicity, how severe or damaging the infection is. So virulence is a measure of how harmful an infectious agent is and its ability to overcome a host defense, their immune system, and cause damage in the host. A highly virulent agent, for instance, requires only a small amount present to cause illness, whereas something that's less virulent will require a larger exposure to cause disease. And virulence can be measured by ID50, which is a representative of the median infectious dose. This is the number of cells, virions, or toxin amount needed to cause infection in 50% of a test group. Whereas the LD50, this is the median lethal dose, which is the number or amount needed to cause death in 50% of the group. Let's take a closer look at ID50 and LD50. So ID50 measures infectivity, how easily a pathogen can establish infection. A lower ID50 means the pathogen is more infectious. LD50 again measures how severe or deadly a pathogen. That's the virulence, so how severe or deadly the pathogen is. And a lower LD50 means it's more lethal. Here we have a graph from the book where the x-axis is the number of pathogenic agents. That could be cells or virions or toxins. And on the y-axis, we're looking at percent death rate. And it looks like in this particular case, you need 10 to the fourth, what is that, 10, looks like 10,000 particles to cause 50% death in that group. And our book provides an ID50 chart for different types of foodborne diseases, and these, it's important to note, are median values. In reality, the actual infective dose for individuals really vary. It depends on factors like your immune system and the route of entry of the pathogen. So one example of this variation is in Shigella. If I look at Shigella, it has an ID50 of about 10 to 200 cells, so it's pretty infectious, highly infectious. Compared to the next one, Vibrio, you need to be exposed to 1 million cells to cause infection. If I look at another example, the two right above that, it looks like a single cell of Salmonella enterica, Cerevar typhimurium. That one, a single cell can cause infection, but the resultant disease has a pretty low death rate, mortality rate of about 1% of untreated individuals, whereas in Salmonella enterica cerevar typhi, it has a much higher ID50, right, 1,000 compared to 1. So you need to be exposed to more cells to become infected, but it has a much higher mortality rate, so as high as 10% of untreated individuals compared to the previous 1% for the other cerevar type. This checkpoint asks, what is the difference between a pathogen's infective dose and lethal dose? So the infective dose, we're looking at that ID50. That's the number of pathogen cells or toxins or viral particles required to cause infection in 50% of hosts, whereas the lethal dose is the number of cells or toxin or virions, those viral particles, required to cause death in 50% of hosts. If I look at which one's more closely related to the severity of disease, the lethal dose is more closely linked to disease severity because it reflects how damaging or deadly that pathogen is. It's also important to note that not all pathogens behave the same way. We have primary pathogens that cause disease even in healthy individuals, such as Bacillus anthracis that causes anthrax, or enterohemorrhagic E. coli that produces Shiga toxin. In contrast, opportunistic pathogens only cause disease when the host immune defenses are weakened, such as in infants, the elderly, or in immunocompromised individuals. So one example is Staphylococcus epidermidis. It's normally found in the skin, but it can form biofilms on surfaces like catheters and then be introduced into the body and can be very harmful, especially if someone is immunocompromised. Similar to that example, microbes can also cause issues when they're introduced in the wrong or to the wrong body system. For instance, E. coli is normally found in the GI tract and it helps us break down certain nutrients and provide certain vitamins. But if they enter the urinary tract or the bladder, they can cause infection as well as disease. And opportunistic infections can also be found when we have disruptions to our normal microbiota. Someone that's undergoing antibiotic treatment for a bacterial infection might kill off the bacteria and allow the normal yeast, like Candida, to overgrow and cause thrush. We also often see overgrowth of Candida and thrush in individuals with untreated diabetes because that leaves a really high concentration of glucose in the saliva. We also see this a lot in individuals with immunodeficiencies. So this first checkpoint asks us to explain the difference between a primary pathogen and an opportunistic pathogen. A primary pathogen can cause disease in a healthy host. I mentioned Bacillus anthracis or enterohemorrhagic E. coli. Shiga toxin, for instance, inhibits protein synthesis, and that can lead to severe bloody diarrhea, inflammation, and even kidney failure, even in individuals who are otherwise healthy. In opportunistic pathogens, these cause disease when the host defense is weakened or compromised, or the microbe gains access to an unusual body site, such as E. coli entering the urethra. For the second one, describe some conditions under which an opportunistic infection can occur. So we said when normal microbes are disrupted, such as through antibiotic use. Another one is when the immune system is suppressed, maybe from chemotherapy, from another illness, or from HIV. Or when microbes enter sterile body sites, right? The blood should not have pathogens or microbes in there, and that might be we might introduce microbes there through wounds or catheters or surgery. Now let's walk through those stages of pathogenesis. These are the key steps that pathogens have to complete to cause disease. And we're going to go through each of them in more detail in the following slides. You'll see there are 4 main stages: exposure, adhesion, invasion, and infection. So that first stage is exposure, also known as contact. This is where pathogens first encounter the host, and this is usually through some kind of portal of entry. Major portals include the skin, mucous membrane, as well as the parenteral route, which means through breaks in the skin. And our book has a nice image or figure that shows the different portals of entry for female and male patients. So again, this is where pathogens can gain entry or access into the body. A lot of these are directly exposed to external environments, such as the eye, nose, or mouth. And the placenta also counts as a portal of entry for pregnant females. This is a portal of entry for the fetus in this case. Of those portals of entry, the most common are through our mucosal surfaces, like the eyes, nose, mouth, urethra, and anus, that make up these respiratory, gastrointestinal, and genitourinary tracts. Of those portals of entry, the respiratory and gastrointestinal tracts are especially vulnerable because we're constantly breathing and eating. For the parenteral route, this can occur through wounds, bites, or needle pricks. And although most microbes cannot cross the placenta, there are a few major groups that can. And Listeria is another example of a microbe, a bacterium, that unfortunately grows really well at cold temperatures like our refrigerators and can also cross the placenta and cause significant harm to the fetus. Some microbes also can infect infants during childbirth, so mothers can cross or pass chlamydia and Neisseria onto the infant during childbirth. Usually these will infect the eyes of the infants, so these can cause disease in the eye, but luckily usually infants are given antibiotic eye drops or ointment right after childbirth or right after birth. And here are those torch pathogens that can cross the placenta. These are the names of the pathogens. You do not have to memorize them, but they're a handy reference. And these are the diseases caused by each of those pathogens. The torch is an acronym for each of these diseases. We can see T for toxoplasmosis, for instance. The others are also acronyms. The O really stands for other. So these are the other diseases that can be caused by these pathogens. After exposure or contact with a host, now the pathogens have to attach to us, to attach to the host cells using adhesins. And these might be found on bacterial fimbriae or flagella or capsules. They might be found on eukaryotic cilia or the viral spike proteins. And this process is known as adhesion. This is how they stick to us. Some microbes can even form biofilms like the one shown here below. That helps them anchor to tissues or medical devices like catheters and also helps them resist immune attack. You might have heard that drinking cranberry juice can help with urinary tract infections. And the reasoning behind this is that E. coli uses adhesins on their fimbriae to bind to the cells that line our urinary tract and to the bladder cells. Cranberry juice, though, contains plant polyphenols. Specifically, they are proanthocyanidins. These bind to the bacterial fimbriae and block adhesion. So this allows E. coli to be flushed out of the system before infection can develop. All right, so far for the stages of pathogenesis, we've seen exposure, also known as contact. Step 2 was adhesion. Now we have our third step, invasion. So once attached, now pathogens can invade deeper tissues to spread and evade or hide from our immune system. Some pathogens, for example, can secrete exoenzymes or toxins that can damage our tissue barriers. So one example is shown here by the book. Helicobacter pylori produces urease, and that neutralizes our stomach acid and allows them to penetrate our mucosal lining. Regarding our third step, invasion, there are also some bacteria that are intracellular pathogens and can invade our cells directly. For instance, we have some bacteria. Let's say that this is a phagocyte. So this is one type of immune cell that ingests microbes like bacteria and chops them up using lysosomes. So some bacteria can survive inside these phagocytic cells by escaping that vesicle called the phagosome before the lysosome, remember that organelle is responsible for releasing digestive enzymes to chop things like bacteria up. So they can escape this process and survive inside the cell and continue to divide within their cell. So finally, after invasion, pathogens can multiply and cause tissue damage. And this growth and multiplication of the pathogen is known as infection, our last stage of pathogenesis. And that takes us to the end of our second video. We're going to pause there. And in the next video, we're going to start with infections again and talk about the different types of infections.